You just have to try. If it doesn't spray, you will get stuck.
Already finished like the spray amongst the dust from just like measuring and preparing the way. I wanted to ask, since the vault is being used to use the site, is that a new equipment? Okay. I was wondering if this temperature is
So you want to make sure it comes to face. If not, you can use it.
All right, can everybody have their seat so we can get started? All right. It's my pleasure today to introduce Dr. Brian Lovett. Brian is an insect pathologist at the USDA and Cornell University. It's a dual appointment. He received his his bachelor's degree at Michigan State University, where he worked a little bit with Ned Walker. He moved on to get his PhD at the University of Maryland with Raymond St. Ledger. And I assume you're going to be talking about some of your dissertation work. He did a postdoc at West Virginia University with Matt Hansen during COVID, so we had a purely COVID postdoc, which was documented quite a bit on Twitter, actually. But for his some of his work during his PhD, he published a paper in Science under the age of 30 and received the AAAS Newcomb Cleveland Prize in 2020 in recognition of that work. He's also very active in the Entomological Society of America, recently serving as program chair, which pretty much requires a sabbatical to do. So he's one of the people who organized the entire program for all the 3,000 to 4,000 attendees. Yep. It's quite the undertaking. And he also has been very active in the Entomological Society of America's science policy program. So welcome, Brian.
Thank you very much. Well, thank you, everyone. Thank you for the wonderful introduction, Anne, and also the invitation. I'm so pleased to be here with this wonderful department of such productive folks. I'm really excited for my meetings later, and I hope that you guys are ready to hear a lot about fungi. And so I'm just going to jump right into this. My talk is very broadly titled because you will see I have lots of interests that will span lots of different domains, and so I also have objectively far too many slides for my time slot, and so we are going to run through this. So initially, I want to talk about what it means to be an insect pathologist to me. And insect pathology, and I think really all of our fields, we should consider them in this way, but it is a particular field where you have to be interdisciplinary. You have to think about multiple fields to really understand what's going on in a system when you're considering an insect that's becoming infected with another microbe. And so in my case, I really sort of span two primary domains, mycology and entomology, and I'm always trying to apply to sort of pit the one field against the other. And so one of the things that I often will struggle with is when I'm trying to understand something mycologically, then there will be a gap in our understanding of the insect. And the same will be true in the other field. And so I'm constantly sort of trying to put these puzzle pieces together, but I think that there are some really fundamental natural history gaps that could really help this type of interdisciplinary work. And so I'll try to highlight that throughout my talk. And so to get us in the mood for insect-killing fungi, I'm going to start with arguably my favorite fungal pathogen, Mesospora cicadina. This is a specialized pathogen of periodical cicadas. This has been known for a very long time. This is a sketch of the beautiful structures that this fungus can form on the cicada host when it was described in 1919 by Speer. And this is a cicada that I found out in the field. And what you may be surprised to learn about this cicada, which is clearly not doing well. You can see the back part of this body has been replaced by fungus. That cicada is still alive. After I took this photo, it flew off with this plug of fungus on it. And so this fungus, instead of engaging in the typical mode of transmission that all the other fungi in its group uses, which is essentially launching spores as a cannon to infect the next insect host, it stays on the live host and manipulates the live host to move it around the environment. And so this is a really unique mode of transmission that involves a really intimate understanding and manipulation of the insect host by the fungus. And so this is something that in my postdoc at West Virginia University, I was really fortunate to work on it. We actually had the emergence of Brood 10 while I was there as well, and so there were lots of opportunities to collect specimens and to think really deeply about this pathogen. And one thing that I really learned working on Mesospora cicadina is that we really have to consider the context of the host to understand the system. And so I'm going to highlight two things about cicadas that are really unique and important for the success of the fungal pathogen. The first is that periodical cicadas, they're underground for a really long time, 17 to 13 years. Annual cicadas too are also underground for some years. They just come up every year. They're not synchronized in the way that periodical cicadas are. And so this pathogen, Mesospora, already has to somehow be able to infect every 17 years when that opportunity arises. And so this is already a pretty unique challenge for this fungus. Our understanding of how it infects them is it actually is waiting for them at the surface of the soil, and when the cicadas emerge to wait for that soil temperature to reach the appropriate temperature for synchronized emergence, that is a period where they should be in direct contact with the infectious spores of this fungus, and they probably are becoming infected as nymphs, even though we see the infection as adults. This is our current understanding of this paradigm, but the further that these cicadas get underground, the less we understand about what is actually going on with them and how many of them are infected with our fungus. And so this aspect of the cicada lifestyle is not only a challenge for the fungus, but also an opportunity. They're sitting at that space for a set period of time where it could actually infect. The other thing you're probably wondering is, why is this cow doing this weird thing in the corner of the slide? The idea here is that there's an old joke among physicists that if you want to optimize dairy production in cows, first you have to imagine that cow as a sphere. I think we do a similar thing with insects, where we may simplify the insects into whatever we learned in our physiology class, or sort of a broad concept of particularly hemocoel just being blood that's kind of sloshing around. If you've ever popped open an insect, it's much more complicated in there, and insect to insect it's very different. And so we have to understand this unique context for the insects that we're considering to understand what the pathogen is actually encountering inside of that insect host. And so I would encourage you to not simplify your insect into a ball of hemolymph with cuticle around the outside. And so what's unique about cicadas? What is the thing that everyone knows about cicadas? Yes?
They like to scream.
They love screaming. And the way that they scream is by clapping together these ribs. This is a slow motion video of this happening. And so you see they're pulling these ribs together, and that rib makes a very quiet snapping sound. But what's interesting about the cicada is the entire back half of the body is replaced with air. And that's a resonating chamber that takes this very small sound and turns it into what I think is justifiably described as screaming. And so in the context of our ball of hemolymph where things are sloshing around, that is not the environment that this fungal pathogen is encountering inside of the cicada. It is encountering basically a pocket of air. And so here's a diagram which is actually showing how this works, and you can see literally basically from the thorax on, all the organs are adpressed to the top of the body, and it's just air, particularly in the male cicadas, although there is a smaller pocket in female cicadas. As eggs develop, it kind of starts to become consumed. And so this quote that I have here is from a very early description of this looking at the development of Mesospora inside of the periodical cicadas from 1929. And one of the things that they commented in the paper is that unlike all the other fungi in this group, Mesospora does not need to seek the air to sporulate. It does not need to emerge from the host to sporulate. Instead, it accomplishes this without ever leaving the animal. And I think the way that it does this is by finding a pocket of air already inside of the host. And so this physiology, this sort of quirk of how that resonating chamber works and how they use that to communicate with one another, this is something which dramatically changes the mode of infection and the opportunities for this fungus. And so we conducted a modern look at the cytology of Mesospora, and we were able to see similar development, but even in this cross-sectional picture, you can see the Mesospora is actually encroaching upon that basically open air within the cicada, and that is the environment that it's growing into. It's really not even interacting with tissue very much, or the hemolymph in any capacity. And in our understanding of this fungus, and in this study as well, we confirmed, it is limited to the back half of the cicada's body. It is not moving through the body. You're not finding it in the anterior portions of the host. And so, curiously, this has been described as a genital pathogen, because it obviously is replacing the back half of the host, and ultimately those parts of the host body will slough and fall off. But in her cytology, it actually left the genitals undigested. So a little quirk of that study that we found. And so this is showing on the right just the general development of that fungus as little bodies into these little packets of spores that ultimately form that sort of plug that I showed you on my first slide. And so this is, I promise, the simplified lifestyle diagram of Mesospora. I'm going to quiz everyone on this in the class after this, and so pay very close attention. But I want to highlight a couple of things here. First is that the cicada is emerging, is molting, and then as an adult, we see the development of symptoms. And so this fungus is actually capable of navigating that molting cycle. And I think that's really important for understanding the opportunities for infection. I think probably a number of insect pathogenic fungi are able to navigate this phase, but we don't really understand that too much. But this is one where it's built into our understanding of the life cycle. Once the cicada develops for about a week, the abdominal rings will actually begin to slough off, reveal this fungal plug that's infectious, and that cicada will just spread fungal propagules all across the branches and trees and everything that it's walking across, presenting an opportunity for direct infection. However, this is not enough for Mesospora. It also manipulates the behavior of male cicadas to further transmission. And the way that we can understand this is through our fairly intimate understanding of the mating behavior of periodical cicadas. When a male cicada screams, the female cicada, if she likes that scream, will flick her wings. And male cicadas will respond to this flick really intensively and will use that as a cue to mate. What we see is that male cicadas, which would normally not flick their wings, when they're infected with Mesospora, they will respond to calls by flicking their wings. And so this shift in mating behavior in male cicadas results in males trying to mate with an infectious plug. And so there's this direct manipulation of host behavior. I mentioned this sort of flicking sound, the flicking of the wings. I just want to show you how attractive this is to male cicadas. And so if you synchronize it to their call, they will literally walk into their death in a snapping finger. Very attractive, something that this fungus has actually been able to manipulate in this host. And this is not a weird example that I pulled out to show you how weird fungi are. This seems to be a theme in these Entomophthalales. We see a similar manipulation of mating behavior in the fly pathogen, Entomoptera musci. Entomoptera musci kills the host, positions it at a high location through a course of events that we typically describe as summit disease, and then this will launch its spores outward to infect hosts. But even still, it is secreting a volatile that encourages males to mate with the cadaver. This is really fascinating, and I think the more we dig into this, the more we're going to understand this is a common approach for entomopathogenic fungi, but this is a great example where we need to know things about the mating behavior of the insect hosts to be able to implicate manipulation of mating. And there are lots of insects where we really don't have a good idea of what behaviors we would expect to point toward it and say, this is manipulation. So working in this group, I really was pulled into this broader field of insect behavioral manipulation. And so this is an article that we put together where that Mesospora life cycle diagram came from, where basically we're just trying to break down all of these different categories of manipulation that we see in fungi. And so we put together this figure, which we affectionately refer to as our subway map, where you can look at basically the traits of the fungus or the traits of the host, understand which type of manipulation you're dealing with. And so I'll walk us through a couple. With Mesospora, the host behavior absolutely facilitates transmission. The spores are not discharged by the fungus, and the host is alive during transmission. And so we would consider this to be active host transmission, where effectively the host has been recruited to transmit its own pathogen. A very dark, fully zombie situation that we're dealing with here. In the case of Ophiocordyceps, the zombie ant fungus, the one that Last of Us was inspired by, this follows a little bit of a different path where the host behavior just toward the end of death does certainly facilitate transmission. The spores are discharged by the fungus, but the host is dead while fungal transmission is occurring. This is one where, because the host is sort of guided up to a high point in the canopy, we refer to as summit disease. And there are other fungi where they don't seem to be engaging in these manipulative behaviors, but we may not understand their manipulative behaviors yet. And so that one is a little bit of a gray, non-manipulative category, but I think that manipulation clearly is beneficial to the fitness of these fungi, and they seem to, across many different evolutionary groups, develop these skills and use similar skills to modify insects. And this is not something which is only limited to insect pathogenic fungi. They're an example of viruses that cause summit disease. They're an example of trematodes that will cause ants to climb and perch onto grasses. And so this is a broad behavior, specifically the summit disease, sending insects to a high point to die, that many pathogens have zeroed in on. And so I put together this article where we proposed a broader paradigm for understanding the summit disease manipulation. And the two behaviors that we put forward for this were sleep in adults and molting in juveniles. We think that these are two behaviors that capitulate more or less the gamut of summit disease behaviors that we see. In the case of molting, there is even a phase when particularly lepidopterans are about to molt called wandering. When they're infected with certain insect pathogens, there is a wandering phase toward the end of life. And so there seem to be these behaviors broadly in insects that may be just triggered by the pathogens. And what's interesting about these particular behaviors that we've highlighted is that sleep is something that is fundamental. You need sleep as much as an insect needs sleep. All insects have to molt. And so these are things which are kind of fundamental chinks in the insect armor that once a pathogen locks into these things, the insect can't just stop sleeping or can't just stop molting. And so these are really fundamental aspects of biology too that we think are really compelling. These pictures here are just showing a solitary wasp taking a nap. Here are some Drosophila that I snuck up on at night. I gave them a little pole to perch on to see if they would actually perch. I took that idea from a study that was using little poles to monitor summit disease development in Drosophila. And if you give it to wild Drosophila that are uninfected, you can recapitulate almost fully that behavior. This has to do with lots of really complex behaviors like negative geotaxis and sort of sleep positioning, but those pathogens may be sort of directing that cycle. Okay. So there are a number of ways that we can dig into this. One that I have really leaned into is genomics. We sequenced the genome of Mesospora cicadina. We thought we could just throw some Illumina reads at it and have a genome and publish it, and it turns out it is the largest sequenced fungal genome. I know to some of you folks that are maybe sequencing insect genomes, 1.5 gigabases doesn't seem too big, but this is by far the largest fungal genome that was sequenced, and it is not complete. We're getting close, and we're continuing to refine it, but this was a technical surprise and a challenge for us. We've been working with Jason Steisig's group and his student Sharon Chu to try to understand the broader context of these Entomophthalales fungi. So here is a tree which is showing these insect destroyers, this group that Mesospora and Entomophthora musci are members of. And you can see in this figure the representation of transposable elements and the genome size. And what we found is that broadly in the Entomophthalales, the genome is full of transposable elements that are massively inflating the genome. And so they're not really getting a huge increase in the number of genes that they have, but the genome itself is just getting larger and larger and larger. And this is really interesting to me because one, Entomophthalales have massive cells for fungi. We can actually see a single spore of an Entomophthalalian fungus through the dissecting scope. You can very clearly see the outline of that spore because of how large those cells are. The other thing is that sort of historically, we've used a stain that stains heterochromatin to identify nuclei in these fungi. And it makes a lot of sense that that would be a really useful stain for them because they're absolutely replete with repetitive DNA. And so we recently published a broader comparative genomic study in eLife, trying to understand, in particular in the zombie fly fungus Entomopthora musci, what aspects of the genome are related to this modification behavior. And so we were able to sort of substantiate and sort of show the massive inflation of genome size and how it sort of does not trace the number of genes that are in the genome. And we were also able to survey for genes that are involved in circadian rhythm. One thing that a lot of researchers that are looking at the manipulation of insects have zeroed in on is that pathogens continue to maintain their own circadian clock within the host and are using that to enact certain manipulative behaviors. And so this is something where we sequenced the genome of this pathogen and are just baseline trying to understand what's there and how might it relate to some of these really interesting characteristics. A major challenge with doing comparative genomics on these is the massive inflation of the genomes and these transposable elements just inserting themselves everywhere. And so this will be an ongoing challenge, but it's something that we're ready to tackle because at my center, we have the world's largest collection of entomopathogenic fungi, RSF. And so we have this wonderful resource for conducting comparative genomics against these fungi. We're taking inspiration from the I5K initiative and using our USDA scientific computing resources to try to collect all the data that's being sort of more or less haphazardly generated on entomopathogenic fungi, put it into a single resource where we can quickly provide comparative information and also identification of entomopathogenic fungi. Whenever people will publish on taxonomic papers on these fungi, they'll deposit sequences into NCBI, and we want to go through and mine those and be able to have a centralized resource specifically for the entomopathogenic fungi that are in this collection. And so we will be taking resources that are already available, but we'll also be going through that collection and identifying fungi which are either under-sequenced or maybe should be in the collection but are not, and trying to target them for the collection. So this is work that we just a couple of weeks ago brought on Kelsey Scott as a postdoc to conduct, and so she's going to be doing a lot of really interesting work trying to set up these tools and these resources and take a first pass at this database. One thing that's really important to us is that every year the number of genomes that are sequenced increases. And so we're not trying to catch up, we're trying to create a tool that will allow us to add in more information and be much more responsive. And so this is something that Kelsey is really well trained for, and we're excited to see the work that she does. Another aspect of this work is actually identifying and getting these pathogens. And one strategy for doing this, which I have been able to leverage really successfully, is using social media to identify fungi. And so Anne had mentioned my Twitter. I was fairly active on Twitter before its downfall. But I remain active on iNaturalist. And these are places where someone can have no idea what they found, but it can make it to you. And so this is an example that I'm showing where a leafhopper clearly has been positioned in a summit disease position. One thing about summit disease in a lot of insects is the fungus will actually encourage them to spread their wings. That's because the fungus emerges from the abdomen. And so if the wings were covering the abdomen, then the fungus would just hit the underside of the wings and wouldn't be able to infect anything. And so they saw this. It was obviously a weird position for the hoppers. They were dead. They posted it online, and they were like, I have no idea what this is. A colleague of mine was like, I think Brian might know what this is. And it turns out this is a pathogen that had not been described from Ohio. And so I was like, yes, please send it to me. This is how I think you should collect it. This community scientist, she packaged these samples up way better than any of my professional scientists. It was amazing. I literally was like unrolling them from like napkins, and I was like, this is incredible. And so we received them very quickly. We were able to get a permit for them. We were able to get them over. And I was looking for this pathogen. And so I've been describing this sort of cannon that they create. And you can see here, the fungus has emerged from just outside of the intersegmental membrane, and all these little dots are spores that are going to be launched off from the host. And so I was trying to cultivate that fungus. And I was like, yes, I want to see which one it is. I have a suspicion of which one it is. These Entomothriales, they have become so specialized to their hosts that even if your profession is to culture them, it does not always go very well, and some of them we still cannot culture. And so unfortunately, this was one that we could not culture. And so knowing that, whenever I get a specimen, I try to send it as many places as I can. So I try to get some genetic data on it, I try to culture it, I also try to do histology on them, because these are all things where even if my culturing fails, I can still learn something about what's going on in the system. And so I sent these leafhoppers off for histology. I sort of foolishly sent off early in infection, middle in infection, and late in infection individuals. All of them looked like this. They were absolutely full of these Entomothriales cells, these really massive purple round protoclasts. And so this sort of confirms to me it is the fungus that I thought it was, and it gives me a little bit more information about it. But I was looking at my plates, and I noticed there seemed to be another fungus growing instead of the one that I wanted to grow. And so I went back to these histology slides, and I realized that there's another fungus in this photo. You can see interspersed with the massive cells a normal-sized fungal cell. These are typically these would be referred to as yeast-like symbionts, and these were also throughout the insect body. We actually use a differential stain where you can really clearly see the sugars on the outside of those fungi are present in both of these. And so knowing that, I went back to my cultures and I was like, is this fungus, am I actually able to grow this fungus? And I was. We were actually able to see it growing on the hind wing that I put into Grace's media. I was able to transfer it to a fairly complex media that has milk and eggs and every other nutrient that I have in the lab. And I was able to grow it. It takes months and months to grow. It is a super bizarre fungus. But the reason that it's bizarre is because this type of fungus is a fungus that used to be a pathogen and was recruited as a symbiont to support insect health. This is something in plant-feeding insects that is seen time and time again. There are also particular insect groups. The one that comes to mind right now are bedbugs that actually have specific organs to house fungi like this. And so this is a known life cycle, but I would say for the most part, it's understudied. And one of the reasons why it's understudied is because it's very hard to culture these because they've been highly specialized to not only their host, but a particular organ of their host. But I was lucky, I was able to culture this. This fungus is very closely related to a lot of other fungi that I cultivate. They grow like molds, they look like little white filamentous fungi. This looks like a pile of gray goo. Something very clearly has changed morphologically for this fungus, even before we've looked anything into it. And so because we were able to culture it, we sequenced the genome of it. We were able to put together a highly complete genome, 97.7% complete, and it is on the smaller side of the genomes. You can see I have a different letter in front of bases here for this genome. And so this one fits a normal size for an entomopathogenic fungus, but is actually a little bit truncated. And so once I had mine, I went to see what other genomic information is available on this type of life cycle. And I was able to pull a number of fungi that are either related or have been implicated in a yeast-like symbiont life cycle. Here I've got two, which are from a plant hopper. This is the one that I isolated from a leaf hopper. Here's a curiously closely related nematode pathogen. And then this one was isolated from a cicada. And this is a sort of outgroup normal entomopathogenic fungus, Tolopocladium inflatum. And when we look at the development, or when we compare the genomes of these fungi, what's interesting is that the number of predicted genes is decreasing in the YLS fungi. They are losing capacity to do things. The other thing that we're seeing is the size of the genome is also truncating. This is actually a little bizarre for fungi. It's kind of a normal thing for bacteria that when they're sort of recruited, that they have this sort of shrink of a genome size. But we're really interested in this because this may be approaching sort of a minimum fungal genome. And that may be sort of the insect domesticating that fungus into that life cycle. The other thing we did is a comparative analysis of the protein content that was present in these fungi. And what's interesting is that my fungus that I isolated from the leafhopper has more genes unique to it than the YLS have in common together. And so what this tells me is that the YLS are probably taking a different path to that life cycle, but they did share just one gene, which is very convenient to have a pool of candidates that is one. And this is a glutamine-dependent NAD+ synthetase. And so this suggests to me that these YLS are all relying on glutamate for currency within the insect host. This is something that we can dig into. The other very tantalizing thing about this gene is that it is described as a bacterial gene. What is it doing in my fungi? And so this is part of a much broader context of understanding the transition of entomopathogenic fungi from being a fungus to a symbiont, and in particular, a defensive symbiont, a symbiont that can protect and support the nutrition of that insect host. And this is something which, when we start looking broadly across insect groups, we see it time and time again. And so in cicadas that were sampled in Japan, 15 of the 24 cicadas that they sampled had a replacement of a bacterial symbiont with a yeast-like symbiont. And you can see here they actually stained the different bacteria in the top there, and you can see where the YLS is actually replacing specific partners in specific tissue. And so there seems to be almost a one-to-one swap, where one bacterial partner is being evicted by the fungus, and the fungus is coming in and occupying their home. And so I'm starting to think much more of the bacteriome as a microbial battleground, a tissue which is very valuable that lots of microbes want to occupy in order to support insect health. The challenge with having a tissue that lots of microbes want to occupy inside of you is that only microbes that disrespect your cuticle are the ones that are available to become your partner. And so it sets up this weird juxtaposition where the pathogens are actually probably the ones which are better equipped to end up in these organs. And that is indeed what we see with entomopathogenic fungi. And so here I'm just showing the bacterial symbionts in Asian citrusillid, and Hodgkinia would be the one that I would expect a yeast-like symbiont to replace in this system. And typically we do see the yeast-like symbionts occupy that central portion of this bacteriocyte, that specific symbiotic organ. And so this has really led me to have a much broader understanding of what pathogens can be. And instead of thinking as the organism itself as a mutualist or a pathogen, I think about the different phases of infection and whether or not those phases are more mutualistic or pathogenic. And in particular, there is a yeast-like proliferation stage where entomopathogenic fungi, to avoid detection by the immune system, no longer grow filamentously. They grow as a budding yeast-like form, which looks exactly like the yeast-like symbionts that we see in these insects. And so what I'm thinking is occurring here is that there is actually a phase of the infection with the entomopathogenic fungi where it's in their favor for the insect to do well, where they're actually behaving almost mutualistically until, cards turn, where insects have an opportunity to lock them in to that life cycle. And I think that's what we're seeing with YLS. And I would love to do that experimentally and intentionally in the lab. And so I'm going to make a really hard pivot here to millipedes. The context here is that when we're looking at these broader systems, when we're looking at the bacteriocyte, for example, a fundamental aspect of understanding what's going on or how that's changed is knowing what's going on normally. And so I think it's really important to engage in just broad description of microbes that are associated with arthropods. And I think this will open up opportunities to notice where those communities have been disrupted or where pathogens are actually having a role that was underappreciated. And so in the Casson lab, when I joined the lab, they had already sort of committed to working on these fungus-feeding millipedes. They're in a subtier order called, or subtier class called Colobignatha. Here we have Matt Casson and Angie Macias. Angie has been doing her PhD work on these fungus-feeding millipedes. And when I had arrived, they had conducted a culture-based study looking at a particular millipede, Brachycybe lecontii, the feather millipede, just trying to look at what microbes can they isolate from it. The challenge with doing a culture-based study is that there are so many biases toward which organisms you can actually culture that we really wanted to provide a bioinformatic sequencing-based approach to be able to compare and understand more fully what's present on these millipedes. And so this was something that I was able to build up and put together in this lab. Just to give a little bit of context for the Colobignatha, they have a wide diversity. They tend to have really beautiful, striking colors. They will form what are called pinwheels on the fungi that they feed on. They have really interesting, complex social interactions where there's brood care and males actually hold onto and protect eggs. The juveniles, when they emerge, actually are unpigmented and are unable to defend themselves until they continue to mature. They also sit very close to the base of the phylogeny of millipedes, and they're understood to be the earliest terrestrial animals and the earliest obligate fungivores. And so from a mutualism perspective, it would be really interesting to know what fungi they feed on, but when the Casson lab started on this, there was one article that was like, I think I saw them feed on this fungus, and that was all we knew. And so the culture-based study and the sequencing study, this is just setting the baseline. What do we understand about the system? And so what we did is we collected a bunch of millipedes from a bunch of different colonies, just to show you. These are the adorable juveniles that sit and sort of occupy under the adults. And we did Illumina ITS barcoding. We used the AMTK pipeline for analysis, which takes a lot of different databases to try to identify which fungi we're dealing with. This was also developed by a mycologist, so as far as pipelines for working with fungi, this is definitely the one that I would recommend. And this is just a broad look at what fungi were there. I'm not going to really try to make any takeaways about this. This is really just the first pass at what are we seeing. Majorly, we see that across sites, there seems to be some consistency. There were no differences between males and females in the populations. This is one thing we were wondering because males were kind of dispersed across, so we were thinking maybe they might hold a different community. And then within colonies, within sort of pinwheels or groups that we collected together, there did seem to be a fairly consistent microbial community with a wide diversity of fungi, very wide diversity of fungi. And this type of sequencing approach, it can allow us to look at some of these fungi that we know cannot be cultured. And so I just want to highlight the types of sort of things we can learn from this analysis. And so here are polyporales. These are these sort of crust fungi that we often see these millipedes feeding on. One that we often see is this one on the right, Flebia. They will be munching on it directly. And so we had an expectation Flebia would be one of the ones that came up for us. And indeed, we see it has really wide prevalence across the group. And so this is something which can allow us to understand broadly their diet, but we're also interested in microbes that are on and inside of them as well. And so this is a very broad view at their diversity. But we are looking in all of this just at one millipede, Brachycybie lecontii, which is sort of limited to the east coast, much more toward the southeast. However, these millipedes are everywhere, and there's lots of diversity of them. They have really fascinating, because of how old they are, ranges where mountains that used to be together, like when all of the continents were a single unit, still follow their ranges. And we are able to sample them broadly across the East Coast and also the West Coast, and those are completely separate populations. And so we received in the Casson Lab National Geographic Explorers funding to go on these collecting trips to all of these sites and collect dozens of these millipedes to conduct the same types of studies. So we've been isolating fungi from them, we've been doing sequencing of them, and we also were doing chemical analysis of them because they also produce novel chemistries to defend themselves. And so we collected all these millipedes. I think just yesterday, published an article on our collecting trip and the discovery of a millipede that we were shocked that we found. And so if you're interested to read a totally non-serious article about it, it's an American entomologist maybe coming to a mailbox near you. This trip really allowed us to sample that broad diversity, but also look at and sort of consider a number of these millipedes. One that I wanted to highlight here is Rhinotus purpureus. This is an invasive millipede in Florida. We typically find these millipedes under logs, where we see fungi sort of growing on those logs. Rhinotus purpureus, we instead found in mulch. This is super concerning because this is an invasive millipede, and pretty much every mulch pile that we found in Florida when we were sampling for it, absolutely full of these millipedes. The reason why Rhinotus is concerning is because the compound that it produces is the compound that poison dart frogs sequester by eating this millipede. And so its distribution and its prevalence in the area, I think, is very important for understanding these broader ecological contexts. And so here I am collecting it from a pile of mulch, I think in a citrus orchard. But because we were able to look at all these millipedes, me as an insect pathologist, I'm looking for fungal pathogens, right? Where are the fungal pathogens of these fungus-feeding millipedes? And there were vanishingly few. We really were not encountering them at all in the field, and it was interesting that we weren't. One really well-known millipede pathogen, and maybe one of the most thoroughly described, is Arthrophaga This is part of those insect destroyer groups that I was talking about earlier, where basically the entire millipede can be converted into those cannon spores. This is one which infects cherry millipedes, and so the host is sort of directed to sit on the top of a log or on a branch, and then it will produce these spores and just rain it down onto the millipedes below. If you have a large population of cherry millipedes, particularly in our area, you will find this pathogen. And while we were sampling, although we didn't find pathogens in our fungus-feeding millipedes, we were able to come across other examples of Arthrophaga in other hosts. And so here on the right is actually one that we collected in California in a different species. Maybe it's the same pathogen, maybe it's a new pathogen that is specialized to the species. We also can troll iNaturalist and see when people post photos of infected arthropods in new hosts. And so one that I find particularly tantalizing is there does seem to be a pathogen in the invasive and highly, highly abundant greenhouse millipede. I would love to be able to wipe that out of our forests and not see it under every log that I look at. And so having this eye toward sort of what pathogens are coming out of them or what we're looking at has always been a really important focus of mine. And the fungus-feeding millipede that we have in Ithaca is Petaserpes cryptocephalus. Here they are feeding on the stem of a shiitake mushroom. It took me an embarrassingly long amount of time to realize that you can just feed them edible fungi. And so we've been maintaining colonies in the lab of these, but when you maintain colonies of insects, you often encounter entomopathogenic fungi. And so here is what remained of our colony, and we found that if we rear the individuals in too high of numbers, almost inevitably they will become infected with Metarhizium, which is, as you will see, a favorite pathogen of mine. And so in the field surveys, we were not able to encounter this, but in my lab, just in a very short amount of time trying to cultivate them, we were actually able to discover and sort of characterize the diversity of these. My suspicion is that this is a millipede specialized Metarhizium, but there's much to look at there. The other thing that I wanted to highlight is some collaborative work that we've been doing on these millipedes, trying to understand these defensive compounds. This is work that we've been doing with Sunny Zheng's lab at Cornell with postdoc Mary Salcedo. Also two amazing undergrads, Matt Huang and Corey Reese, who actually became so interested in this system, he just joined my lab. And they have been doing high-speed filming of the production of defensive droplets by these millipedes. And so let me see if I can play it. And so here's a millipede that we are very gently pressing with a cover slip. And what it's going to do is encourage the release of defensive compounds. And what we've seen, if you sort of look at the top of this millipede, is a clear droplet emerges first, and then a white sticky droplet. This is occurring so fast that if you look at it, it just looks like a white droplet appears. What we're actually seeing is that there's a phased release of the defensive compound, one which seems to wick up the body, and the other one which follows that other compound up the body. These millipedes are incredibly curved, and so having something that could actually move that defensive compound up the body, we think could be really useful for them. The other thing, which we can see in this video, is that the millipede is actually able to pass the defensive compound across the center of the body, to the side where it's been perturbed. That's really interesting because this is a highly costly compound for it to produce, so being able to place it where it wants it specifically, we think would be really useful. This is just one approach to sort of using that filming, but we were digging in on sort of what defensive compounds are there and what might they be doing in this broader system. And so, for the rest of my talk, I'm just going to talk about biopesticide development. Why do we need biopesticides? We need them because chemical pesticides face an uncertain future. We need them because invasive pests continue to arrive and continue to be here. And also, we need to develop the tools for tomorrow today. And so, here I'm highlighting just one example in spotted lanternfly of a native pathogen, which is already providing some level of control. The more we can understand and characterize these things, the more we can intentionally go out and work with them. And so there are a number of ways that we can develop these new pesticides. The first is just by going out and finding them. And so this is a project that I proposed during my postdoc, where we actually went to Christmas tree systems to try to look at elongate hemlock scale, the major pest in Christmas tree systems, and just survey for a fungal pathogen. The first forest that we went to, we saw these beautiful orange dots on the underside of the needles. We were able to bring it back to the lab and characterize it as a specialized scale pathogen, Conoidea rostrata. It certainly infects the elongate hemlock scale. We think it's probably going to have a host range that will allow it to infect other scales. It has incredible biocontrol potential, and we actually just published this work in Mycologia, and they liked the fungus so much they put it on the cover. And so if you're interested in scales or their biocontrol, I'd love to hear from you, and this is something that I want to continue to develop for growers up in our area. What's interesting is this pathogen has been historically described from the US, particularly in whiteflies, but sort of simultaneously, our group and another group noticed it was in a bunch of Christmas tree farms. And so there seems to have been a recent resurgence of this in elongate hemlock scale in Christmas tree farms. We don't exactly know what's going on there. The other thing we can do is we can take fungi that we already know about and we can screen them against pests. And so this is a project that a postdoc in my lab, Elizabeth Moore, has been conducting. She's also been working with technician Greg Biddle, who's there snuggling with Anasandrus maisch. And this is a project which is part of a larger SCRI project looking at ambrosia beetles, and we're focusing in on trying to support New York apples, the ambrosia beetles which might be in that system. And so for this study, we took 3 fungi, Cordyceps, Metarhizium, and Bavaria, and we just exposed these different beetles to these fungi to see how susceptible are they, and at what level, what dose of spores do we actually see these beetles being killed. We developed an assay, a very sophisticated assay to infect these beetles. We put fungi in a tube, and we vortex the beetles in that tube. They become highly, highly infected, but that allows us to get that sort of baseline information of, would this fungus even work under the best case scenario? And so I'm going to show you some survival curves. I'm going to run through these. Anasandrus maisch is on the left, Xylosandrus crassiusculus is in the middle, and Xylosandrus germanus is on the right. If the survival curve comes down to the bottom, the fungus is working very well. If it stays toward the top, it's not working very well, and the darker the line, the higher the concentration. Everybody's with me. So Anasandrus maisch, even at the middle concentration, is being killed. But in the other two, it takes the highest concentration of the fungus to actually kill them. This is a theme that we saw across our work, and so this suggests that Anasandrus maisch may be more susceptible or a better target for entomopathogenic fungi than the other two. This is looking at Cordyceps fumosa roseus, which is admittedly our worst one. With Metarhizium bruneum, we see in Anasandrus maisch, all concentrations, even the lowest concentration, kills all the beetles. In the others, even the middle concentration of this one killed both beetles. In the case of Bavaria, we see a little bit of a mixed bag with the higher concentrations up there, but we are seeing a consistent trend across Anisandrus maisch and the Xylosandrus. What we're thinking is going on with the Anisandrus maisch is that it has a thinner cuticle. When we do that vortex assay, they tend to get dented and beat up a little bit more than the others, and that may be something that allows the fungus to get into the host faster and more fungi to get into the host. So once we were able to see sort of which worked, we wanted to see under a more realistic scenario, could we actually introduce the fungus to these beetles? And so here, we actually treated a bolt with spores, allowed the beetles to sort of walk through that bolt, burrow into the bolt, and then we allowed the beetles 2 weeks to sort of set up their gallery and population, and then we cracked them open to see how successful were they. And in this study, we set them into these containers in a controlled environment with the sort of treated outside for 2 weeks before we cut everything open. And we just wanted to understand, how does development of gallery affect what we were seeing in our sort of fully plate removed assays? And so we had really 2 major treatments. One where we took our beetles, which were vortexed in a tube, put them directly onto an untreated bolt and allowed them to develop their galleries. And we had another where they were not treated, but they could pick up an infection on the bolt. And this work is still ongoing, but on the left here are the beetles that were treated directly, and on the right here are the bolts that were treated directly. And the major takeaway here is that treatment on the bolts results in very low infection, if any. However, these beetles were treated with such a high dose, they all should have died. And so the fact that there are live beetles in here suggests that actually the development of a gallery and the maintenance of the symbionts somehow is protecting them from infection, which is really curious to us. And so one thing that we looked at was, are they picking up enough spores? Is that the problem? And if we look at this, and we actually look at the survival curves, and so here the orange one are the ones where they're exposed to bark, and then this is actually the survival curves from before, just to give you the context of what we saw from direct treatment. They're getting sort of a medium to low dose just walking across the bolt. And so it does not seem that they're not picking up enough to die. And so what we're thinking is that there are these broader mechanistic questions. Are the entomopathogenic fungi susceptible to the ethanol that attracts the beetles to the tree? Are there volatiles by the symbiotic fungus, which are allowing that symbiont, the sort of crop that they are breeding, to actually protect against infection? The other thing is maybe there are other associated fungi that aren't that sort of canonical symbiont, but are still present in the system, which may be providing some sort of protective effect. And we have arrived at the last part of my talk, the last part of my title, and I think my most exciting direction for entomopathogenic fungi, which is developing them as platforms for biotechnology. And I've been talking a lot about the biology of these fungi, but I think this is the first time I've shown you an infection diagram. And so these fungi typically land on the outside of an insect cuticle. Here they will actually taste which insect they're on. And if they like the taste of that insect, then they will continue to burrow and develop infection structures to penetrate that cuticle. This cuticle, to scale, would have to be like a couple rooms up. It takes about 2 days for that fungus to make it all the way through this phase. Once it makes it into the hemolymph, the fungus starts to grow entirely differently. I mentioned earlier this is to avoid the immune system, but this means that up here and down here, the fungus has completely different suites of genes that it's expressing. And so we can take promoters from those genes and use them basically as on-off switches to say, I want the fungus to express the thing here, or I want the fungus to express this gene only when it's inside the insect hemolymph. And so this is the platform, and I describe it as a platform because you can really put any gizmo you want into this insect and into this fungus and have it express it inside of the host. And so I'm going to be talking about one strategy, but I see this as highly flexible strategy for delivering nucleotides, proteins, chemicals into an insect. This is work that I did in the St. Ledger lab at the University of Maryland, and it was generously funded by the NIH. The strategy that I'm going to talk about today is taking toxins from arthropod predators. Here's the Blue Mountains funnel web spider. If you poke it, it releases a little droplet of venom. You can pull up that venom, you can fractionate it, and you can look at all the proteins that are present in that venom. Our collaborator, Glenn King, loves to do this, and he found one which is called hybrid, which has been vetted by the EPA for application in greenhouses. So this is one that we thought had a certain level of safety evaluation that it would be good for this technology. And so when we introduce this toxin under a hemolymph-specific promoter into these fungi, we find in the lab it kills Mycetes faster, it kills them at a lower spore dose. What this results in the field is increased persistence. If it requires fewer spores to kill, even if you have fewer spores alive, you can still kill. We thought this was really promising in the lab, but just because something works in the lab does not mean it's going to work in the field. And so we developed a collaboration with Abdoulaye Diabaté in Burkina Faso to conduct semi-field trials to understand, does this work in the field? So we have a very simple objective. Do we see the same effect, the higher virulence in the field with the transgenic fungus compared to the wild-type fungus? To do this, we had to construct a facility for years. It's like the quote from Carl Sagan, if you want to bake a pie, first you have to invent the universe. If we wanted to make one graph, first we had to build this entire facility. And so here I want to highlight Diabaté, Etienne Dogo, the graduate student here, and we were also able to conduct stakeholder meetings at our facility as well, which is really important for the community. So we built this in the village Sumoso, a malaria endemic village. We were able to collect wild mosquitoes from the area to actually fill our compartments, and this sort of baby picture shows you the breakdown of the facility. It's essentially a really large greenhouse, but instead of having glass, it has a double wall of mosquito netting that allows environmental conditions, for the most part, in, but prevents our experiments and mosquitoes from actually getting out of that facility. Within each compartment, we also had an experimental hut that we were able to put natural vegetation onto as well. This provided an environment where we could really rapidly conduct these studies. And so here is my colleague Etienne. He is sampling for mosquitoes in the compartment. Behind him is a black cloth that we have impregnated with fungal spores. So we literally mix the spores in sesame oil, we soak that cloth overnight, and then we hang it up in a compartment. That was our only intervention, that was our treatment in this system. We would release 100 mosquitoes into the compartment, and then the next day, we would collect every single mosquito individually, and then monitor the survival of those mosquitoes over 2 weeks. And so I'm gonna show you that figure. So in green here, we have the hybrid toxin, the ones that had a oily sheet that had a transgenic fungus on it. In the red, we have the wild type fungus. We marked it with RFP just so we could confirm that our fungus killed that mosquito. And then we have mosquitoes that were just subjected to an oily sheet in blue. And what we see is exactly what we saw in the lab. It kills more mosquitoes, it kills mosquitoes faster. I want to highlight that day 3 is really when this kicks off. That's how long it takes the fungus to get into the hemolymph of the mosquito. So that's when our transgenic fungus is actually behaving transgenically. But we just built this whole facility. We can't just make one graph, right? And so we conducted a suite of other studies trying to look at how combination of arthropod toxins from different predators might have a differential effect. And we were actually able to engineer a strain where one spore kills one mosquito. We also looked at whether or not infection affected insecticide resistance. One phenotype which is really critical for insecticide resistance is called knockdown, and we found that as mosquitoes were becoming sick, it restored knockdown to mosquitoes that should be resistant to pesticides. If we look at blood feeding, even before the mosquito is dead, we've stopped malaria transmission because they lose interest and ability to blood feed. But in our compartment, we could actually establish mosquito populations and monitor the development of populations. And what we found is in 2 generations, the mosquitoes were laying so few eggs that we had collapsed that contained population with the transgenic fungus. In the wild type fungus and the control compartments, those populations were increasing, but you'd walk into the hybrid compartment and there wasn't a mosquito in sight. Really compelling results looking at sort of developmental impacts of our transgenic toxin on the adult mosquitoes. But I have to share with you, I know I'm short on time, but I have to share with you the other thing that we did in these compartments, which is sexual dissemination of entomopathogenic fungi. And for these studies, we treated male mosquitoes by putting them in a cage that was covered with the spores. And then we released them into a compartment alone, and then we released 250 uninfected females into that compartment. We had to have this difference because mosquitoes, they actually have to form a lek. You can see the little white flecks up there. And so the male mosquitoes form a swarm, and the female mosquitoes fly through, and if they want to mate, they'll form a couple and they actually fall out. And so the system allows us to not only set up these populations, but also monitor how many of them have mated, which is really interesting for us. And so we put many couples in the sphere. About 25% of females in this artificial compartment actually ended up mating. And then we did that same individual recapture, just monitored survival. And so the males were treated, the females could only pick up infection through mating. And this is what we found. Males that are treated directly with spores, they die. Not super interested. Females, if they're mated with a male that had the transgenic fungus on it, pretty much 100% of the time she'll die. With the wild type fungus, it's about a coin flip. The reason why I say about 100% of the time is because only 25% of the females mated, so this is really our population that could become infected. And so this is really was really interesting to us. One thing we looked at was actually how many spores are being transferred during mating events. So we actually matched up the mosquitoes and counted how many spores we could recover. And we found that it was about 9 spores. From our previous studies, that is enough of the transgenic fungus to kill. It is not enough of the wild-type fungus to kill, and so that fits exactly with what we saw. One question that we had is, could we treat the mosquitoes and then release them later? And so we did that experiment here, releasing the male mosquitoes after 1 hour of treatment, 6 hours of treatment, 12, 24, and 48 hours of treatment. This figure is a little messy, but the major takeaway is that up to 24 hours, you see pretty much the same effect. And so you could feasibly treat a male mosquito, drive it 24 hours away, and then release that male mosquito to have that effect. And so this is really interesting from an operational perspective, but it suggests that that fungus has a sort of window somewhere towards 48 hours where it can actually infect. This also fits with what I was saying about penetration in the cuticle, where by 48 hours, if it's an infectious fungus, it probably should have committed to infecting whatever mosquito it was on already. We've continued to develop this work by isolating native entomopathogenic fungi from Burkina, so continuing to just study non-transgenic fungi. And one thing that we've been really interested in is the interplay between Wolbachia and the entomopathogenic fungi. And we have found in the lab that these native entomopathogenic fungi, which are quite good pathogens of Anopheles mosquitoes and Aedes mosquitoes, that the Wolbachia is actually protecting them from infection. And when we look at different temperature regimes, if we look at the typical rearing temperature that we would have mosquitoes in the lab, or a more field-relevant temperature, the bad news is the field-relevant temperature provides more protection from fungal infection. And so the interplay between these symbionts, that sort of battleground within the host, is really important to understand for the success or lack of success of these biopesticides. And so I'm looking at this in a really broad context. I see these fungi and the different evolutionary pathways that they can take as having a lot of opportunity for modification. I think of them as platforms where we can think of ideas of things that we want to do and then use the fungi to deliver them. And so these are a suite of biotechnologies that we're continuing to develop in our lab, either looking at effectors targeting molting and circadian rhythm, trying to take some inspiration from summit disease and what pathogens have been telling us they think is a good strategy. Also developing RNAi silencing of host genes. This proof of concept has already been demonstrated in these fungi, but it really has not been leveraged in the way that it could be. Disrupting host symbioses, trying to intentionally change that battleground and the stakes there. And of course, just techniques for routine and rapid modification. So I think I have time for half a question, but I do want to thank my lab members, Greg, Kelsey, and my support scientist who has contributed to everything, Elizabeth and Corey. So thank you very much.
Not half a question, right?
Yes. Yes.
Okay, so I have a couple questions, but I'll ask one of them.
I only have time for half a question.
The
Yeah.
yeast-like fungus
Mhm.
that you find, how do you know that they're always helping the insect? Because sometimes, what if they're having a synergistic effect with the other funguses? The two funguses are mutual.
I think that definitely could occur. There's very little that we know about them, and so I wouldn't rule it out.
Okay.
That's a good idea.
Chelsea?
Hi.
Yes. Excellent talk.
Thank you.
Very fascinating. If I had to narrow it down to just one question,
Uh-huh.
what is the likelihood, or is there a great concern or any control parameters you can use to ensure that these fungi won't be highly likely to host jump to different species?
Yes, so this is a fundamental question that these fungi are a wonderful model for studying because these fungi can have either generalist host ranges or they can be specialized to the point of being specialized to one host. And so periodical cicadas are kind of weird because they have that glaciation, now they're a little bit of a grab bag, but like Entomophthalales, they have one species that they infect. And so fungi do have that capacity, but I would say it exists along a spectrum. And I think there are certain circumstances where having a specialized host would be super useful. And I think there are other circumstances where we could apply a generalized host in a sophisticated way and just target the insects that we want to encounter. In a malaria context, if an insect is coming into your house, it's already kind of violated and would be considered a pest in the first place. And so you can use treatment methods to take sort of generalism and apply it in a sophisticated way. Typically, people do look for specialization and understanding what leads to host switching. It's a major focus in the field, but we haven't cracked that nut entirely just yet. Yeah. Yes?
For like lack of a better or more accurate term, do comorbidities or like other defects in the system make it play a major role in using pathogens to control insects, or is that something that's...
You mean like some of these other effects on fitness?
Yeah, I guess like age, perhaps.
Oh, yes.
Old or
absolutely.
I don't know if there's some sort of morphological deficiency or something.
One thing that I think we do a lot is when we set up bioassays in the lab, we use like very standardized cohorts, and it's often like the most fit or like one specific sex, and then we draw broad conclusions about them. And so certainly I think there's a lot to be done just looking at sex differences and differences in physiology, in particular fat content within hosts, I think is really important for understanding this. But there's absolutely going to be effects on age, status, and also other pathogens. That's one thing that we're really zeroing in on, is how does the sort of pathogen status or symbiont status affect the success? Because maybe we have a colony that doesn't have a YLS, and then we try to expose it to insects that do have a YLS, and there may be some of that competition that was mentioned earlier. Yeah, absolutely. Yes?
The genetically modified mechanism seems very effective. So what's the likelihood that can be implemented in the next 5 years? What's the main obstacle?
I would say that for that technology, the main obstacles are political and regulatory. And that is something that I think is really important to approach in a thoughtful way where you're engaging stakeholders. And while I certainly am involved in the development of that project, the pace of that and how that's developed is being determined by our Burkinabe collaborators and their needs and discussions with their agencies. Sometimes policies across countries can be very similar, and sometimes they are not. And so I'm very engaged and interested in these issues, but for that particular project, I'm not the one that's leading that, and I think the time frame is going to be largely dependent on some of these other non-scientific aspects. One thing that we try to do is really make the science clear and be like, this is the value of using this. If people don't want to use it, that's their call. But I think in a malaria system, people really want to use it.
Sure.
Yeah.
So can you use a similar approach to design genetically modified material for, say, cockroaches and bedbugs?
You could use it for cockroaches, you could use it for bedbugs, you could use it for any number of plant pests. This is highly translatable, and if you find a fungus infecting your host and you send it to me, we could develop a very similar paradigm.
There is a product for bedbugs.
Yeah.
You may modify it.
Yeah.
I think we're going to have to go to the
I did not trip over my microphone.
I was so terrified you were going to.
I'm a pacer. I only pointed at a couple things, but...
Oh wait, there's some chat.
Oh, good chat.
It's actually a beer company.
Yeah, toxoplasmosis is a solid example, but also it's overstated. Devin, can you put
Oh yeah?
On this?
Yeah.
I feel like not a lot of people say that about poultry.
Maybe.
It's not a school shot.
Yeah! Yep. Yeah, that's what we're talking about. Absolutely. Thanks, Devin.
But thanks so much for
Yeah, absolutely. Really great chatting with you this morning, too. Yeah, reach out.
Yeah.
I had a buckwheat.
I was wondering if you ethanol too.
I thought you would be.
Yeah, because I'm like sitting here, I was taking notice, like, what about if we can use ethanol baited some logs as bait and then try it? But
We actually have a specific...
Bye everyone.